we have Miriam Klein Flügge from Oxford, who's a neuroscientist here. I'm just hand over to Miriam right now, if I may. Thank you.
Great, thank you so much for the introduction. It's great to be here and to get to know people who work on a range of things related to mental well-being. So I'm very excited to present our recent work to you, which hopefully will come up, everyone can see. That's perfect. So yes, I have recently started my own research group here in Oxford, and I really work at the interface between mental health research and neuroscience. So for any economics or other backgrounds in the audience, bear with me when I talk about the brain for most of this talk. But I do think the brain really matters for our well-being, and hopefully everyone agrees on that. So what I will be talking about is how we can maybe see in the brain whether there is a relationship between the connectivity of the amygdala and other parts and participants, or our human mental well-being. So just to give you a little bit of background, I'm sure many people here will be familiar with the fact that it's been really, really important in the neurosciences and elsewhere to try and understand what predicts mental well-being. And it will be predicted by all sorts of environmental factors, but also by factors that are in the brain that we can find in our biology. And so one way to look at the brain in a healthy human is, of course, to use non-invasive methods such as neuroimaging. And so many studies have by now come out where we use neuroimaging markers to predict participants' mental well-being, and sometimes it's in cohorts that are suffering from psychiatric disorders, and the aim might be to predict disorder severity or maybe symptom profiles. And so what is observable in many of these studies, and I've just picked two examples that I'm going to show you briefly, is that usually these predictions, how well is someone doing in terms of many dimensions this could capture, usually rely on a really large brain network. So here are two examples of studies that have come out, and I could have picked others. They're both using resting state functional connectivity, meaning how do brain regions communicate with each other. And then they're basically trying to get the highest prediction accuracy to say, is someone struggling with depression or not in these two examples. And so you can see that they actually base their prediction on a really, really large brain network. I mean, even as a neuroscientist, it is impossible to really read into this graph what's going on, because it involves so many different nodes in the brain. All around the circle, you can see different brain regions, and then their connectivity relates to that prediction of whether someone is likely to be depressed or not. And so here on the right, a similar idea with a slightly different approach, where depression was sort of subdivided into these 4 subtypes. It was a very influential paper. There are also problems with it that I won't go into the details of, but it's certainly also tried in a similar vein to predict depression with brain markers, and it did so incredibly accurately. So the prediction accuracies were around 80% in these studies, meaning they're very accurate. But as you can see, it's really hard to interpret which parts of the brain are really the crucial parts of the brain that contribute to this prediction. And so we wanted to take a slightly different approach, because we realized this actually ignores what we know from a lot of literature, both that is based on animal models of disorders such as depression, as well as in humans, dating back to PET work looking at depression. So we do have some priors of the networks that might be important, also based on their function. So we wanted to use a large number of healthy individuals. So it's of interest to this audience. If you can any of us, we're all on the spectrum, but within the healthy range, hopefully. And so this is the Human Content Project. Some of you might have come across it. So really, mental well-being in this entire group is just varying on a very subclinical basis. As scale, we do not have people with a diagnosis of a mental disorder, because they were recruited such that they are healthy. But we are really asking, can we find very specific anatomical circuits that we can interpret in terms of the function that they observe and relate them to mental health damages in this health? So it's a quite different approach. We're not aiming for the largest possible prediction accuracy, but rather we're aiming for the largest possible interpretability, so to speak. So that also in the future, the aim could be that you could actually imagine in people who struggle with particular mental disorders that we could actually alter these circuits, which you can imagine is really hard to do in circuits as large as the ones here. How you actually kind of try and maybe rebalance these networks. So we're trying to see if we can find smaller networks that are still maybe useful at predicting some amount of variance. So as in the introduction, it already became apparent, this work is going to focus on the amygdala. It's by no means the only region we should be looking at, but it's a very good candidate to start with. And so there's many reasons why it is one of the key regions for regulating expression emotions, basic neuroscience literature. We know that when you remove it, which sometimes is done in patients with epilepsy, for example, it alters our fear and our very healthy fear and anxiety responses. This is also, worked by multiple different groups. And then we know that the amygdala has an abnormal heightened metabolism in depression, which is PET work. And we know that is also abnormal when you do specific tasks that are related to emotion processing. For example, how you process emotions of other people's. And then finally, from a more neuroscience-y stance, if you may allow, if we look at the networks, and I won't explain them in a lot of details, the amygdala is a really, really interesting interface. So you can, here in the middle, this is where the amygdala is, and you can see it has these various different nuclei, and some of them have very direct projections to the brainstem. So to just highlight a few of these connections and why they're important, some of them will allow us to do fight and flight responses. So they are very autonomic, very direct, immediate responses to our environment. But then the amygdala also has these connections to regions in the prefrontal cortex. And so we know that that's really important for guiding behavior for much more cognitive and demanding process. So it's a really interesting interface of very fast, but also very cognitive, slow thought through response. And so that was the motivation for why we focused amygdala in this study. The way we measure connectivity here, there's multiple ways to measure it in the brain, and we chose one particular method, which basically just asks, if I look at the time course in a specific region measured over time, as shown here, and I look at the time course in another region measured over time, I can just correlate them, and that gives me a measure of how coupled their bold response. So this is the fMRI signal we measure, how coupled they are. And so the more coupled they are, the more likely it is that there's a strong anatomical connection between them. But what we do not know from this functional connectivity measure, which is purely correlation measure, is whether it's a monosynaptic connection, so whether it's a direct pathway. It could be a bidirectional, it could be a bisynaptic, so it involves multiple synapses. And so here is one way to show that functional connectivity really reflects anatomical connectivity, so really the brain circuits and their connection. And just to guide you here, basically on the left we see, in this case, this is to the monkey brain, the left and the right hemisphere, and you can see that you often get these this connectivity between the left and the right hemispheres in these off-diagonal elements. And the best way to see what they are is when you look at the difference here. This is basically when the hemispheres have been separated, which is why it had to be done in an animal. So they basically just cut through the hemisphere connection. So the left and the right side of the brain do no longer talk to each other. And you can see that if we then measure functional connectivity between the hemispheres, which is these off-diagonal lower left and upper right box, the connectivity has basically disappeared. So it's a good marker for how anatomically connected regions are, even if it's not a direct marker of that, which we can't have in humans. There are also some challenges with the sort of study that we wanted to conduct, which is that the amygdala is very deep inside the brain, as you see here on the left. And so what that means is that because we measure our signal using the MRI scanner and the head coil is right around the head of the participant, we often do not get a great signal from structures that are more deep inside the brain. We get a great signal from more superficial cortical structure. So there are some things to consider. And then the other thing that we need to consider in terms of not just the signal we get, but also the anatomical structure within the amygdala. Often it's referred to as one unit, as one structure in the brain, but actually it's not. The amygdala has a lot of different nuclei, and the sort of connections that I just pointed out earlier, for example, are a very different part of the amygdala. It's the central nucleus, for example, that has all these endocrine and autonomic connections that will make you react very fast, or related to different neurotransmitter release. But then you also have the connections to the prefrontal cortex, and they tend to be actually with the lateral and the basal. So different parts of the amygdala actually are not quite the same, organized the same way. And that's something that maybe we need to consider if we really try and understand what these do, because likely if their connections are very different, then their function is also very different. Okay, so the question that guides this work is then, does amygdala connectivity, or more precisely, the connectivity of its precise nuclei, relate to dimensions of mental well-being? How connected the amygdala is in the brain tell us something about how well someone is doing in real life? And so that's what we try to answer using a large group. And so my book really has these three parts. First, for those who don't know that much about the brain, I will have to bore you a little bit about brain anatomy and talk to you about how we managed to parcellate the amygdala into these component parts that we think really matter, that do very different things. And then in the second part, maybe more of interest, I'll talk about how we looked at different dimensions to people's behavior, including their life stability or negative emotions, sleep problems, and with anger. So those are the sort of behaviors we will be talking about related to their mental well-being. And then we're bringing these two parts together in the third part, which is really answering the main questions. Can we identify amygdala connections in the brain that then relate to these behaviors that predict whether someone has more or less problems with sleep, for example? Okay, so coming to the amygdala parcellation, we basically put each participant in our cohort, which initially was 200, but later we increased it actually to almost 500. We have these measures for the entire brain of where they are connected to, and we can average that across all our participants to make it more robust. And then we get something like the group connectivity. You can see it has a lot more structure in it already. And so when we use that group connectivity and just look at where are the regions that strongly connect to the amygdala, on average across all participants, so this does not yet look at the differences, you can see that the amygdala connects strongly to itself. So just to guide you, this is the amygdala here we're looking at. But the amygdala also connects strongly to regions we know it should connect strongly to. So for example, here on the left, we have the hypothalamus, neuroendocrine responses. Here we have the ventral striatum, which some people may be familiar with in terms of reward processing. It also strongly projects to the medial surface of the brain here and the temporal lobe. So that's a good check, because if that wasn't the case, our data wouldn't be very meaningful. We know that that is the region that the amygdala projects to. That's a sanity check. Our data is reflective of some meaningful structure in it rather than just noise, which some people do say it is a problem in the amygdala. We might sometimes measure mostly just noise. So that was a good comforting thing to see. But then what we were really interested in is, can we use these amygdala-voxel connectivity profiles to parcellate out these different parts of it? So we used a hierarchical clustering algorithm to ask for any two voxels in the brain, are they very similar to each other in terms of their connectivity profile? And if yes, they should end up in the same cluster, in the same nucleus, or are their connectivity profiles to the rest of the brain very different from each other, in which case they should end up in different nuclei. And so this is what you can see here. We basically found this clustering solution. So in all of these panels, you can just see the amygdala. It's just that we're looking at different kind of views. So if it's easy, maybe easiest to just maybe focus on this middle view here. Where you can see it as if I cut through my skull like that. So you're looking at it from the front, and you can see the left and the right here. And you can see that in each hemisphere, we have 7 different nuclei. And some of them are really small, like the dark blue one. Some of them are much larger. And so all of these are features that emerged naturally that we didn't impose on the clustering algorithm. And you can also see that they're actually quite symmetrical, not perfectly, but quite symmetrical across the hemisphere. Again, this is not something we imposed, it's something that emerged. So those are all things that told us, well, probably there really some meaningful variants we're looking at in these data. And so we can then give these nuclei a label just to make it easier to show you the results later. And so one of the nuclei that I mentioned earlier is really important in terms of its connection to the brainstem and therefore autonomic fast responses in the central nucleus, so in dark blue, which is very small, and which previous studies in humans have not actually been able to parcel out because it is so small. So we think we have quite good data quality. So we're no longer going to refer to it as the one amygdala, but rather different nuclei within it, which we know may have different functions because of their connectivity profile. Okay. All right, so this is just to show you, because that's something that, at least in the neuroscience world, and I'm sure economics as well and everywhere else, the replication crisis that people really care about, just to show we can replicate this nucleus parcellation in other data sets. So we have some replication data sets. Here's another 200, here's another 100. And this is just to show you that they're all very similar if you compare them in space. But I will skip over that in the interest of time. Our parcellation looks robust. So here you can see some other regions in the brain, just highlighted again, just for visual illustration in different colors. The colors have no meaning other than to just show you different parts in different colors. And so what these regions all are is regions that we know are really strongly connected to the amygdala. So we know they talk to the amygdala all the time in everyday life. And so those are the ones that we are particularly interested in this study, because it would make sense if the regions that the amygdala talks to a lot are maybe changed as a function of someone's mental well-being. And so some of them you might have heard about, even if maybe not with these names. So the DPOG and VLPOG is periapoplectic brain. They're really important for fighting life and rest and digest responses. So for up and down regulating the body in a very basic manner. The substantia nigra here in black is a structure that is really important for releasing dopamine, which is one of the main neurotransmitters in the brain. And then we have the other neurotransmitter system here. One is serotonin. So the wrapping nuclei are important for serotonin, the locus ceruleus for noradrenaline. So it's all the neurotransmitter systems, which we know are sometimes tipped in one way or another in different mental health cohorts struggling with mental health. So those are the regions we're interested in terms of how they talk to the amygdala. So those are our regions of interest, if you like, that we defined a priori before we started this work. And here you can just see the different regions plotted along X. So these are all the labels of the regions. You don't have to remember them, but they're basically all these regions that are highlighted and shown to you here. Some of them are in the cortex, which is shown here. Some of them are subcortically, really deep inside the brain, like the ones shown here. And I'm just showing you how each one connects to these 7 amygdala nuclei subdivisions. Yeah, and so you can see, for example, the reward structure nucleus accumbens projects most strongly to the central nucleus. And now you can interpret color again as strength of connectivity. And so you can see that some other regions, for example, if you take area 25, which we know is very is highly altered in depression, it connects really strongly to these cortical and basal nuclei, which we know are very connected to the medial prefrontal cortex. But for example, not so strongly to the central nucleus, which is the one that protects. So you can see there's a lot of structure in here. But what we're really interested in now is how does this relate to the individual and how the individual is doing. So now we just have our connectivity profile in each person. So for each person, we have 196 of these cells, right? So it's 7 by 28 in this matrix. And we have that for each participant. And we want to know, does it tell us anything about how well they're doing? That makes sense. That's the anatomy part. Okay. It will become a little bit more easy to understand from now onwards, because we're going to talk about behavior. So the second part was of the question was, well, how can we best and most in a most sensitive way look at how well people are doing? And unfortunately, like with any data set, we were bound by the sort of markers that were required as part of the Human Connectome project, which is the data set we're using here. So we could only look at behaviors that they had asked their participants about via questionnaires or task measures. We can't just ask them anything in retrospect, because it's a big data set that has been acquired elsewhere. And so we basically went ahead and just looked at any markers, which in the end was 33, which were available as part of the Human Connectome project, which is what HCP stands for, which capture these aspects of emotional well-being. And so we were relatively lenient in what we included. So we included, for example, questions on the NIH Emotion Toolbox, the Pittsburgh Sleep Index, but there were no explicit measures included of psychiatric disease. So, for example, if you're familiar with the DSM-5, exactly like a depression score index, we didn't include those. And I'll explain that why. But so what you can see on Y, and I haven't spelled them out, sorry, on X, is these 33 markers, all of which capture something about how these individuals are doing. And what I'm plotting on Y is then the results from a factor analysis. So we're now letting the factor analysis group these variables in the most meaningful way to extract different dimensions of mental well-being. And so we were told by the factor algorithm that there's 4 factors in these data. And so you can see here what the different factors load on most highly. So for example, in this blue factor, the highest loadings, which I've shown with these numbers, are emotional support. That's the highest loading here. The 14th one is emotional support. And then positive effect, friendship, life satisfaction, and self-efficacy. And there are other high loadings. I just wanted to give you a flavor of what goes into this fast factor. So you can already sense, right? Emotional support, how much emotional support you have, how much friendships you have. It's sort of like a general, it has a social dimension to it. How connected are you to people around you? And it has this general, how satisfied are you in life dimension to it. Yes?
I'm so sorry. What is in blue?
So blue are the loadings from the factor analysis. So into the factor analysis, we feed all these questionnaire items from the animation motion toolbox, picture click index, etc. So there's 33 scores that feed into the factor analysis, and the factor analysis gives us basically a weighted sum of these different inputs. So it says that it makes sense, given the variance in the data, to group together these different measures that are emotional support, culture effect, friendship, life session. So these are just weights. It's saying in this factor, the strongest contributing weight is emotional support, but these other ones also strongly contribute. But then it says there's another factor in the data, which is sort of independent, or not completely independent, but somewhat independent from the first one, which loads onto a different group of questionnaire items, right? So again, these are just the loadings. So larger weight means larger influence of that particular score in the factor. And so, for example, here the gray one loads really strongly on fear, which was measured in two different ways, affective and somatic, on sadness, on anger, and on neuroticism. So it's a very different kind of flavor to this factor, right? This factor really captures if someone is struggling with various negative emotions. It's not perfect, because maybe you would want to separate sadness from fear, but it sort of groups generally negative emotions. Yeah. Then we have a factor which solely loads onto sleep. So problems with sleep, the measure we had in the data set were how many minutes does it take you to fall asleep? Have you got trouble falling asleep within 30 minutes? How many wake-ups at night? Sleep quality in general, do you take sleep medicine? So you can see that just the green loadings are not very high on the negative emotion, they're not very high on the social or emotional support, but they're very high on these sleep questionnaire items. Okay, so what we will refer to as the sleep factor, it captures something about the quality of the participants. And then the last one is probably the sort of one that I'll talk about the least, because it's a little bit harder to interpret, but that is just what the factor analysis gives us. And so it loads on hostility, rejection, which is again a social element to it, but on the variable as well, but also on physical aggression and loneliness. So it's probably something to do with anger and rejection, maybe not the most easy to interpret. But those are the 4 behaviors that we will be using to look at the brain, basically. Is there something that in the brain tells us if someone is struggling with negative emotions, and is that going to be a different part of the brain than if someone is struggling with sleep, for example? Does that make sense? Okay. Again, for replicability, we could, I'll skip through this, but you can just replicate this factor analysis really robustly in a data set that includes 100 people scanned at a different scan site, 400 people, and then even 1,200 for which we didn't have all the MR data, but we did have the behavioral data. Or the MR data wasn't sufficiently good enough quality. So it's very robust, these factors. And so we have specified what we want to look at in the brain and what behaviors we want to look at, and now we want to bring them together. And this will be a little bit technical, so bear with me. So basically what we now have is we have these behaviors. So in blue, you will now always see the behavior that was the first factor, life satisfaction. So we might have someone's dimensional score for life satisfaction here on the y-axis, and we have someone's functional connectivity in one edge of the brain, right? So here, for example, I'm showing between the amygdala and a part of the prefrontal cortex. And so the question is there any relationship between the two? We have functional connectivity, and we have the dimensional score of how life satisfied someone is. And you can see here there's a very small relationship, there's a relationship, but it's not very big. But that's what you have to get used to when you look at brain data. It's not going to be 100% accuracy here. But we can ask that same question for all the different edges in the brain that we want to look at, which you might remember were 196, right? So here is 196 of these values, and the one that has a little star on it was the one I had just displayed. So now, what these values signify, what the colors signify here, is how strong the correlation was between the functional connectivity in that brain circuit and the life satisfaction score, right? So it's correlations, again, but correlations with behavior now rather than just within the brain. And so we can basically then ask, well, so we have 2 cohorts. One is 400 participants, and the other is about 100 participants. And so we want to know if we train a network on one cohort, can we predict the other? And so what we want to know is, if we look at that second cohort, do we see similar relationships, for example, with life satisfaction? And so that's what you can see here. And, if you just judge by eye, and we will get to the proper stats, this is just to get, ballpark, whether there's any chance it might work. You can see some connections that are strong predictors seem to be the same connections. There are some eye elements that maybe are comparable, but not all of them. So is that more similar than we would expect by chance? So can we predict from one data set how the other data set scored it? So this is the same for the other for the other three behaviors, so for all four behaviors. And what we can ask without Mr. Method then is the similarity we would expect by chance lower than the similarity we have in our actual data set? And yes, that is the case. So our actual data is a lot more similar, even if it's not perfectly matching between the top row and the bottom row than you would expect by chance. So we think there's some meaningful relationships to these four behaviors in our amygdala connectivity data. And I realize I'm going through this relatively fast, so if you have questions, and we can maybe get back to it at the end. So we can do that in multiple robust ways. So we can also use half of the data in each participant to predict the other half of the data in the same participant, or we can do an across group kind of prediction. And in each case, what we can see is that these patterns of relationships between brain networks and their well-being scores are more similar between groups than we would expect by chance. And all that tells us so far is there's really something in the amygdala network connectivity that relates to these scores. Otherwise, we shouldn't be seeing any relationships between different cohorts or between one half and the other half of our data set. But what we really want to know is, can we predict someone's score based on their brain connectivity, right? And so the most valid way to test that is using out-of-sample predictions. So we train, as I mentioned, our weights on one cohort. So I train how strong does this brain network versus this other brain network relate to life satisfaction based on one cohort, and then I try and predict the behavioral dimensions in another cohort. And the predictions are weak, but in 3 of our behavioral dimensions or mental well-being dimensions, they are significant. So using just all amygdala connections for the time being, and that may not be the most sensitive, but for now we've used all our amygdala connections, we can predict with some portion of the variance if someone is more likely to be very satisfied in life or not, if someone is more likely to have problems with negative emotions or not. Sleep doesn't look great, but for anger it works as well. So what we don't know yet, though, is the similar kinds of networks important for these predictions, and that is kind of part of the really important question here. So what do the networks look like that lead us to these predictions? So what I did here is I asked, what is the smallest number of connections in the brain out of our 196? I may not need all of them to get a good prediction. What is the smallest number that gives us a significant out-of-sample prediction? And those are our out-of-sample predictions with the very smallest number that just reaches significant. So by definition, these are barely significant. They're the smallest network that gives us a significant predictions. And here are the associated anatomical networks. And I'll walk you through these. So in the middle, you can see the 7 amygdala nuclei in each case. So here we have our network for life satisfaction. Here we have our network for negative emotion. Here for sleep and here for anger. And then what you can see on the circumference of the circle are in light gray areas that are in the cortex. So this is the insula FOP. And then we have the medial part of the prefrontal cortex. And then we have some lateral prefrontal cortex here. And I can, remind you of these if you have questions about them later. But so what is then in darker gray are the subcortical regions, so the ones that are deep inside the brain, kind of buried here. So these are like the nucleus accumbens, the reward structure, the PID that I told you is important for autonomic responses, etc. These neurotransmitter systems. So these are all the really deep brain structures. And what you can see is important for life satisfaction when we look at this network is that stronger connections with all of these cortical regions, and so stronger is here shown in a continuous line and weaker in a dotted line. Stronger connections with, for example, the insula and the amygdala, and also the prefrontal cortex, other parts of the prefrontal cortex and the amygdala, gives people a higher chance to be more satisfied in life. So leads to a higher life satisfaction score. Of course, there's no causality relational, as I have to be careful with my language. But then you can see that with subcortical structure, it tends to be that weaker coupling there is associated with a higher life satisfaction score. And here the other anatomical networks are quite different. So that is something we're really happy about. So when you look at someone who struggles with negative emotion, you're more likely to be able to predict that from subcortical networks. And you do not necessarily need a lot of cortical connections. And similarly, the connections for the other two behaviors are also quite different. So this is just to say when we use much more complex scores, for example, someone's total depression score, and remember these are healthy participants, but we can obviously still get them to fill out the questionnaire that asks what is the depression score. We actually struggle a lot more to predict that from such a small brain network And maybe that's not surprising because we know depression has a lot of different dimensions to it. It's not a very well-defined disorder. So that's just to show that maybe having these behavioral dimensions that are specific helps to relate it to very specific brain networks. And then that is my summary slide. So we are able to parcellate the amygdala into 7 subnuclei, which we think is really important when you know about the anatomy of these circuits. And we can measure the resting state data of these individual nuclei with other parts of the brain that we know are important. We have parcellated behavior into functionally meaningful subclinical mental health dimensions, which we can then relate to the brain connectivity. And so we think that this anatomically informed approach really means we can get at these smaller networks that A, are functionally interpretable, and B, we might even be able to alter in mental health disorders in the future. And so with that, I just want to say all the data is available, all the analysis code is available if you want to check that. And we are looking at something similar in relation to stress right now. So that's just to flag that there will be a paper coming out which is more related to stress, but I won't cover it today. It's related to the hypothalamus of the brain. And so this is the group. So thanks to my group, the group that I worked in as a postdoc, and the people who contributed to this work, and of course also to the funders and to you for your attention. Thank you very much.